12 modern application of microbial genetics. So if you remember, we talked about Watson and Crick's discovery that they could identify the DNA structure and in 1953, and after that a lot of major discoveries and advancement happened in the field of biotechnology and genetic engineering. So we use a lot of tools such as restriction enzymes that we're going to talk about them. They are like molecular scissors that we use it to cut the DNA fragment at a certain sequence or in a certain area of the DNA, and we use plasmids. If you remember, those small circular DNA that they both restriction enzymes and plasmid, they actually are derived from the prokaryotic cells in order to manipulate the DNA and make recombinant DNA, and we can make like the products that it's the human desire. And in farming, in agriculture, in like medicine, pharmaceutical areas, and a lot of different fields. So then we're going to talk about polymerase chain reaction, or PCR, that you probably heard about this important technique, so that we can use an enzyme. They call it Taq polymerase, which is a DNA polymerase, very heat resistant DNA polymerase. They actually extracted it from the bacteria that lives in the hot spring, like Yellowstone hot spring. So they extracted this heat resistance Taq polymerase or DNA polymerase, and now they use it in a technique we call it PCR in order to amplify the genes or the fragments of the DNA that they want. Also, a lot of applications in medicine, for example, genetic engineering of the genetic engineering that they do introduce genes into the bacteria to be able to produce the products that they need. For example, they used to have to get insulin from the pig or cow, like pancreas. And you can imagine how many pancreases we need in order to get enough insulin. But now easily they found the genes that can make this protein, this hormone, and then by inserting this gene into the plasmid, and then they introduce it to the bacteria, so they can make and produce a lot of a high quantity of this hormone, and then they use it in the pharmaceutical area. Also vaccines and a lot of biodegradable polymers, we can just produce it by using like genetic engineering. And so microbial genetics have actually re-evolutionized medicine, in diagnostic, in industrial, in the food industry, agriculture, farming, and has a very important impact on our daily life. So this is a thermocycler or a PCR machine, so that we're going to talk about it. And we use the PCR in order to amplify the number of millions of copies of that DNA of our interest, and then we can use this DNA in order to do like further analysis. For example, in this case they can use it for identification of the bacteria that they call it Chlamydia trachomatis, that it's not easy for us to culture this bacteria actually in the lab. So, and it's also you don't want to deal with this bacteria because it's the STD disease. And so you easily, by using like extracting the DNA and then by using the PCR technique, you can see that if the patient is infected with this bacteria or not. So very important technique that we're gonna talk about it. So biotechnology is a very important field that we use the natural biological systems such as plant, animals, human cells to create the products or achieve the goals that we desire. So, and genetic engineering is actually on one of the techniques that we have in the biotechnology, that in this technique we're going to insert the gene of interest into an organism. So we're going to introduce like a foreign genes from another organism to the cell, for example to the bacteria, and then we're going to produce like a transgenic organism that they are genetically modified. So for example, we make like plants that they are resistant to the certain insects or fungi or pathogens, or we introduce like the genes to the cows that they can make more or better quality of milk. And we use this genetically engineer organism for our desire and in order to make like the products that we want. So let's talk more about genetic engineering. So imagine that this is a human cell, and we could identify like the gene that can produce insulin. So what we're gonna do, if you remember the plasmid, those are like a small circular DNA that we have it in some bacteria. So we're gonna use the restriction enzyme, and restriction enzyme, they are actually natural enzymes that the bacteria they have, and they use it as like a defense mechanism to, for example, it can chop up or break down like imagine like the DNA of their uh, antagonist bacteria or viruses when they attack them. So we're gonna cut the plasmid with the restriction enzyme, and there are so many different restriction enzymes such as ECR1, BAMH1, HIND3, and so on. So you can choose which enzyme you want. So you cut the plasmid with the certain restriction enzyme, and then you're gonna cut the gene that can produce or encode insulin in the DNA. And so then because so we call this like a sticky end, so because it has like a overhang area, so these two parts are capable to attach together and anneal together. So then the DNA, the foreign DNA, it's gonna come, and with the help of an enzyme that they call it ligase, it's like a glue, it's gonna glue and stick this DNA or this part of the DNA to the plasmid. So part of this DNA comes from the bacteria, part of it is from the human. So that's why we call this a recombinant DNA, because it's the combination of like two different organisms, or for example two different species. Then they're gonna transfer this recombinant DNA to a bacteria, imagine E. coli or another bacteria, and then they're gonna transfer this plasmid there. And so if you remember, the plasmid can be replicated completely separately from the genomic DNA, so it's completely independent, and then it start to make like several copies of itself in the bacteria. So the bacteria doesn't care if it's a recombinant DNA or if it's own DNA, so it can amplify this plasmid over and over. So then we're going to have a colony of the bacteria that they all contain the in the gene that can make and produce insulin, and then we can extract this insulin and then we can use it for our own purpose. So there's a nice video here, I highly recommend to watch it. So this process, we call this molecular cloning. So so we're gonna, if you remember, cloning or clone means like you make like a specific or the same genetic information, like the colony, that they all have the same genetic information or the same identical genetic information. So during this process we're going to make a clone, so that, like for example the bacteria, that they all contain the gene for production of the insulin. So this is like a big picture and like a very nice summary of the genetic engineering process. So let's learn a little more about the restriction enzymes. If you remember, I said restriction enzymes, they are like natural enzymes that the bacteria they have, and they produce it in order to break down the DNA of their antagonistic bacteria or like pathogens that they can attack them. It's just like a defense mechanism for the bacteria. So imagine if we cut the plasmid at a certain sequence that restriction enzyme can identify it. So then it's gonna produce like an overhang part, so we call this sticky end, and this sticky end is very easy. And then if you cut the, for example, the gene of your interest with the same restriction enzyme, because these strands are complementary to each other, so they can attach together, so they can anneal together. And sometimes the restriction enzymes, they don't make this like overhangs area, so they can just cut the DNA at a certain sequences, in a blunt end, so there is no like a sticky end or overhangs layer, so we call this like a blunt end. So we're gonna cut the plasmid with the same restriction enzyme, and then we're gonna cut that insulin gene with the same restriction enzyme, and then with the help of the ligase they can glue them and attach them together. So this is a nice example for you. Imagine EcoR1. This is one of the restriction enzymes that is very common to use. So it can identify a certain sequence, and then it can cut between guanine and adenine. So it's gonna cut like our DNA at a certain area, and so it makes like the sticky end. And then if we cut these like the DNA with EcoR1 as well, because these strands are complementary to each other, so they can actually anneal and attach together. So the plasmid that we use, they call them also vectors, in order to introduce like a new genes or new genetic information to the organism. So these plasmids are actually modified, so they have to have certain characteristics in order to be able to be used for transformation. So PAC19 is one of the very popular plasmid that we use. So they have to have a certain region, so they add certain region to them that has different restriction enzyme sites. For example, you can see these are like all the enzymes that we can cut the plasmid here using this enzyme. And then they have to have a gene for antibiotic resistance, because we have to find a way to see that which cells they actually obtain the plasmid through the transformation, or which cells they don't have the plasmid, because we don't need the cells that they couldn't obtain, or they couldn't get the plasmid. So let me show you in a like a simple drawing. So imagine this is our plasmid, and now we want to insert like the gene of the interest, which is like a insulin gene. So this is like a fragment that can make the insulin hormone for us. So imagine they're gonna cut this plasmid with EcoR1 restriction enzyme, and then they're gonna cut this gene in our human DNA with the same enzyme. So then they gonna this part of like these fragments gonna attach to the plasmid. So we have to have certain ways that to be able to see if this plasmid exist in our bacteria or not, or if the plasmid contains our insert, our desired gene, or not. So so one way to see that if the bacteria could get the plasmid is by putting the antibiotic resistant gene, that in this case we call it ampicillin, in the plasmid. So imagine now this is the plasmid. Now we transform it to the bacteria. So we're gonna use some techniques to introduce this like a plasmid into the bacteria. So when we do the transformation, some of the bacteria may get the plasmid, some of them may not get the plasmid. So some of them they don't have the plasmid, some of them they have the plasmid. So what we're gonna do when we culture the bacteria after transformation in our media, so this media has ampicillin on it, so it has so it has antibiotic. So just the bacteria that could get the plasmid, they are able to grow in this media. So they have like that gene resistant genes to ampicillin. But if the bacteria they were not able to get the plasmid, so through the transformation, so it's not able to grow in this media. Another thing is that some of these plasmid may have the insert, may have the insulin gene, some of them they may not. So in order to see or to identify just the bacteria or just the colonies that they contain the plasmid that got the insert, got the insulin gene, so we're gonna use this receptor genes. One of the most popular one, they call it LAC-Z gene, can produce an enzyme that they call it beta-galactosidase, and this beta-galactosidase is able to break down a product that they call it xgal, and they actually add it to the media, so they call this product xgal. So it has a big name, but you just need to know like their abbreviation. So they already added this chemical to the media. So if the gene, this LAC-Z receptor gene, is attached or is complete on the plasmid, is able to make the enzyme, and then it's going to break down the xgal to a product that it's kind of make it like a blue color. So the blue color colonies, they are actually the bacteria that they don't have any insert. But if the bacteria has an insert, so it contains like that insulin gene, because the insulin gene is now attached to the plasmid, so it's going to interrupt and break down that lack Z gene, so the gene is not anymore functional, so it's not able to produce the product to, or the enzyme to break down the xgal. So xgal is not gonna break down to the like end products, so they're gonna stay white. So they call this blue and white screening method. So the blue bacteria are the bacteria that their plasmid doesn't contain the insert, so that's why the LAC-Z can make the enzyme and can break down the xgal to a blue product. But the white colonies, they are actually the bacteria that they have the plasmid, and the plasmid contains the insert and has the insulin. So that's why the interruption in the LAC-Z receptor gene cause that this gene cannot produce the enzyme to break down the xgal, and the bacteria gonna stay as white. So, and you can see here a nice also cartoon that how this genetic engineering work. So this is our foreign DNA, this is the plasmid. We're going to cut both of them with the same restriction enzyme, and then, for example, this restriction enzyme can make a sticky end in both DNA fragment and in our plasmid. Then we're going to glue them together using ligase, which is the enzyme that can do ligation. And then we're going to transform this plasmid to the bacteria, and then they're gonna grow the bacteria on a media that contains an antibiotic such as ampicillin, and also contain xgal. So if the bacteria doesn't take up the plasmid, is not able to grow in the ampicillin resistance, in the media that has like ampicillin, so because they don't contain the ampicillin resistance gene. So if the bacteria takes up the non-recombinant plasmid, the plasmid that doesn't have the insert, so the LAC-Z gonna stay intact, so it's gonna make the enzyme that's gonna break down the xgal, and then you're gonna see these colonies as blue. But the ones that you are looking for are the bacteria that they take up the recombinant plasmid that could interrupt the LAC-Z gene. So then the LAC-Z is not able to make the beta-galactosidase, and then when you don't make the enzyme, so we cannot break down the xgal in the media, and the colony is gonna stay white. So white colonies are the ones that they contain the recombinant plasmid. Let's talk about some of the steps that we use in order to transform, or in order to do the transformation of the plasmid to the bacteria. If you remember, I told you like transformation is one of the technique that the prokaryotic cells use in order to get the diversity. So we're going to use these characteristics of the bacteria to do like the cloning, or the making the recombinant like DNA and cloning them. So imagine this is the bacteria, this is our plasmid in the environment. So this is like that recombinant plasmid that we have, and it also has like the gene of our interest. So just show it with a different color. So naturally, the bacteria, through the transformation that we talked in previous chapter, they can engulf these plasmid and bring it inside their cells, and then they can amplify it or replicate it. But in order to make this process more efficient and quicker, so we artificially make this bacteria more competent to be able to uh, do the transformation in a more efficient way. So we're going to use some chemicals that make the cell membrane kind of more loose or more competent in order to obtain and in order to get the plasmid in their environment. Or they use electrocomponent bacteria. So these are the bacteria that for example, they even can use electricity that makes some small pores in them, that it helps them in order to gain the recombinant plasmid much easier from the environment. They even can use conjugation. It's not very common, but also they can use, if you remember, conjugation. We have a donor bacteria, we have a recipient. So naturally, by the pili or the bridge between those bacteria, F positive and F negative bacteria, they can exchange the plasmid. And we have the transduction, so they can use viruses, if you remember bacteriophages, in order to introduce like this recombinant plasmid from one bacteria to another bacteria with the help of viruses. So now you learn about how the genetic engineering and making recombinant DNA works. So let's talk more about some other techniques that we use in biotechnology for production of the products that is our desire. So I'm going to continue in the next video in order to make it easier for you guys to follow.